All right, welcome back everyone, good morning. So we're going to continue looking on at the innate immune system, and today we're going to focus on inflammation, so just building on what we were talking about in the last couple of lectures. So I think we'll start off with the attendance pin. Actually, we're a few seconds early, I think. So we'll just give it 5 more seconds and it should work. So the pin is 6083. That's 6083. All right, so the learning outcomes for the lecture today is that by the end of it you'll have an understanding of inflammation as both a protective acute response, but also the potential harmful causes that we can see in certain pathologies in specific conditions. You'll also be able to understand the features and phases of the acute inflammation, as well as understand the acute phase response and the acute phase proteins. And then we'll continue to have a look at phagocytosis. So this is something that we obviously touched on in the last lecture, but we will expand on that today. So just as a little bit of a recap, you remember that the immune system is comprised of various layers of defense. So initially we have our anatomical barriers, so our skin, mucosa, and the microbiome. Should these fail, we can then rely on the immediate innate immunity. So this is our antimicrobial peptides. This is ready for action at all times. It's ready to go. Should that fail, we can then rely on the induced innate immune system. So this will usually sort of take place, about 4 hours to 96 hours after it's been called upon. And this is our innate immune cells, so the cells that we talked about last week. So these are mobilized upon detection of a pathogen. And should that fail, we can then bring in the big guns, so the adaptive immune system, which is a little bit slower, but it's much more specific. And the benefit of that is it provides us with long-lasting memory, which is obviously the goal of a protective immune response, so that if we should encounter that infection again, then we'll be able to fight it off quicker. So continuing on with that summary, we've covered how the immune system can recognize pathogens via pathogen recognition receptors, but also the involvement of pathogen-associated molecular patterns as well as damage-associated molecular patterns, but also that a recognition of a pathogen and also associated tissue damage can initiate this inflammatory response, which is so important in the immune system. And obviously that's what we're going to focus on today. So this is a little bit of a recap, but it's the picture that you've seen before. So it's quite helpful to kind of think of this in terms of, like a little scratch on the finger or a cut on the finger. So here we've got a little bit of a thorn causing an injury. So you get that kind of bacteria getting in through the cut. You're getting a slight infection. You're calling on the immediate innate immune response, so within the first 4 hours, because it's like immediate. And hopefully the pathogen will be recognized by these preformed soluble effectors that we have, but also the resident effector immune cells. And then, with any luck, that pathogen will be eliminated, and there'll just be some very minor tissue damage. But if it's not, we can then bring in the induced innate immune response. So this will be activation of our resident immune cells, so activation of our macrophages, and we'll start to get inflammation, potentially fever, and the induction of the acute phase response. We'll get soluble effector molecules being formed as well, and then, obviously, hopefully, we'll get elimination of the pathogen. But failing that, we would then move on, obviously, to the adaptive immune response, which is something that we'll talk about next week, I think. So you can see just the different stages here. So we've got our resident effector cells potentially detecting those bacteria, and then we're getting recruitment of the induced innate immune cells into the area, and we're getting this inflammation of the site of infection and damage. So what exactly is inflammation? So we've already talked about the pathogen or the pattern recognition receptors on our innate immune cells, and these are able to respond to PAMPs on pathogens or DAMPs that are associated with damage from an infection. And this really is a trigger for inflammation. So inflammation is classed as this localized accumulation of fluid, plasma proteins, but also leukocytes, so the white blood cells. And this is initiated by that physical injury or the presence of pathogen. And it gives you these characteristic sort of traits, which is redness, swelling, heat, pain, but also loss of function. And so you can see that on this diagram here. We've got a splinter, we've got an introduction of bacteria into the body, and basically these resident or like sentinel effector cells will actually detect the bacteria. We'll get some phagocytosis, but they'll also release chemoattractants as well as vasoactive factors. And what that will do is it will kind of increase permeability of the vascular endothelium. It will recruit cells, so neutrophils, if you think about that, and they'll cross the endothelium by this process known as extravasation, which is something that we'll talk about later on. So the neutrophils will then migrate to the site of the tissue damage, and this is known as chemotaxis. So they'll be following, in a sense, like a breadcrumb trail that is leading them to the site of damage and leading them to the site of inflammation. And this is mediated by chemokines. And then once we get sort of recruitment of neutrophils and other phagocytes, we'll get phagocytosis of the pathogens and hopefully clearance. But that is kind of the steps to inflammation. So you're getting this swelling and recruitment of immune cells into the site of tissue damage. And you're getting all these characteristics that are associated with it. So this is a diagram that I think we looked at in the last lecture. But essentially what we're going to look at now is really the role of macrophages and neutrophils, because these are those kind of early innate sensor cells. So if you remember, we have these inflammatory inducers. So this can be PAMPs, so the bacterial lipopolysaccharides, or the ATP or urate crystals in the instance of DAMPs. But it's essentially these pattern recognition receptors that we have on the surface of our innate immune cells. So macrophages would be a good example. These PRs are being induced by the PAMPs or the DAMPs, and then ultimately we'll get this phagocytosis. Remember we talked about the transcription factor NF-kappa B and the important role that that plays in the immune response. So this will induce the expression of various inflammatory cytokines that will help to coordinate the immune response and the inflammatory response. So remember that the cytokines are those all-important signaling molecules. And these will help to kind of coordinate, but also recruit phagocytic cells, so our neutrophils, into that infected tissue and mediate all the aspects of inflammation. And then the recruited neutrophils will sort of propagate that phagocytosis of the infected area and help to clear and isolate any pathogens that are within the tissues. So any invading microbes will be recognized and removed by phagocytosis, and this usually occurs pretty immediately after a pathogen enters the body. It will be recognized promptly by these resident phagocytic cells. And if you think back to the last lecture where we were looking at the various types of phagocytic cells, you can remember that we've got our macrophages and our monocytes. So the monocytes are the precursors to macrophages. Some of our granulocytes are also good at phagocytosis, in particular the neutrophils. And then, of course, we have our dendritic cells. But remember that the dendritic cells, the phagocytic event is slightly different, because obviously with dendritic cells we want to preserve the material so that that can be presented to the adaptive immune response. Where more often with neutrophils, the aim is really to clear and remove that pathogen and to break it down. Now looking at the pattern recognition receptors, so this is a little bit of a refresher as well, but obviously remember that in the case of macrophages, we have these pattern recognition receptors on the cell surface, Also remember that the macrophages are really kind of the key phagocytic sentinels in the tissues. So macrophages are resident in the tissues, so they're the first cell that will likely encounter a pathogen. And they have this whole host of pattern recognition receptors on the cell surface. This can bind and recognize bacteria or pathogens, yeast, for example. And then we start to get this formation of a phagosome. And we'll sort of get engulfment of the pathogen. This will then form, basically a phagosome, which will then fuse with a lysosome to form a phagolysosome, which you can see here. And then we get this increasing acidification of the phagolysosome, and we get destruction and removal of the pathogen. So just to recap, that obviously we have these PRRs that will recognize the various molecular patterns on the microorganisms, and then this aids in that phagocytic event. And then obviously that broken down material will be expelled, and hopefully the pathogen will be cleared. Now, when it comes to phagocytosis, as I mentioned, obviously the key outcome is that we want to kind of clear that pathogen and also remove it and destroy it. And this is a diagram that we looked at in the last lecture. So you can see here the bacterium being bound by the PRRs. We're getting engulfment, and then we're getting the formation of the phagolysosome and degradation. So obviously that's one key route. But you also want to remember that these pattern recognition receptors, when they recognize these bacterial components, we then start to get the synthesis of inflammatory cytokines. And this can help to boost that inflammatory immune response. So you need to think that we get these activation of the PRRs, we're getting this increased activation of NF-kappa B, that transcription factor, so we're getting transcription of various inflammatory cytokine genes, which will lead to the release of these inflammatory cytokines and then propagate that inflammation. So looking at phagocytosis in a little bit more detail, so you can see here we've got our bacterium or our pathogen. It's been bound by our PRRs. We're getting these actin microfilaments that are forming these pseudopodia, which are actually engulfing, so surrounding the bacteria, and then completely engulfing it until you get the formation of this phagosome. You can see here that the pH of the phagosome is initially about pH 6.2, but then when it fuses with the lysosome here, we can see that we get this acidification, so the pH will drop, and obviously that's quite a hostile environment. We're going to get destruction of that ingested material, or in this case the bacterium, and then the products will be released from the cell. So we've got a whole range of sort of bactericidal agents or antimicrobial agents that are being produced and released by these phagocytes once they've uptaken a pathogen. Now the majority of these will be released into that phagosome, and we'll get the destruction and the clearance, but some may actually sort of be secreted from the cell, and that can be quite toxic to the host cells. Now this table is just showing a whole range of antimicrobial mechanisms of various phagocytes. So we're looking at macrophages and neutrophils. So you can see that we've got this acidification, which is obviously quite important in the phagolysosome. We're also getting the production of toxic oxygen species. So we're getting superoxide anion, hydrogen peroxide, again playing an important role in the phagolysosome. We're also getting nitrogen oxide. We've also got various antimicrobial peptides. So we've got our cathelicidins and our defensins. So when it comes to these, obviously there are lots of different constituents, but I think it's just generally important at this level just to, be aware of cathelicidins, defensins, and histatins, that kind of thing. Not necessarily to go into the specific details that are on this slide here. And then, of course, we've got our enzymes that we talked about. So our lysozyme, which plays a really important role in digesting the gram-positive cell wall. But we also have competitors, such as lactoferrin. And these are only secreted by neutrophils. And so what these aim to do is actually sequester away really essential nutrients from microbes. So this makes those nutrients inaccessible to microbes and therefore sort of hinders microbial growth. And lactoferrin is one that actually sequesters away iron. And then one other important factor to be aware of, which I already mentioned or touched on earlier on, is that macrophages are our resident phagocytes. So they're resident in the tissues of our cells, but neutrophils are the phagocytes that are actually recruited. So the macrophages will recruit the neutrophils, and the neutrophils will follow via chemotaxis that kind of breadcrumb trail that will take them to the site of inflammation. Now this slide is touching on the respiratory burst, and this respiratory burst is what is essential for destroying and clearing pathogens that are taken up by phagocytosis, specifically within the phagolysosome. So once we get activation of our phagocytes, we get this promotion of the respiratory burst, and we start to get this assembly of this NADPH oxidase, which you can see here, and it's this multi-component sort of unit here. And once we get sort of the maturation of the phagolysosome, we'll get activation of this NADPH oxidase complex, and we'll get an increase in oxygen consumption. So we'll get production of the superoxide anion. This will be then converted to hydrogen peroxide, and this will be done by this enzyme here, this SOD, this superoxide dismutase. And then we'll get a whole range of reactive oxygen species being derived from this hydrogen peroxide. But we'll also get production of reactive nitrogen species. So you can imagine this makes a really hostile environment within that phagolysosome, which will degrade and destroy any sort of pathogen that has been phagocytosed. But can you think what else might be happening during this respiratory burst? So we're making quite a hostile environment for the pathogen. But as we mentioned in the previous slide, some of the contents of the phagolysosome can actually be secreted. So what else might happen during this respiratory burst? Yeah?
Neighbouring cells will be killed or damaged?
Yeah, exactly. So it's a really fine balance between fighting the infection, but also preventing damage to our own tissues or to neighbouring cells, like you just mentioned. So for that reason, inflammation really has to be tightly controlled. So ideally, inflammation will be localised, but we need to make sure it doesn't run away, because otherwise we risk damage to our bodies, as you just said. So we were kind of looking there at what goes on in macrophages, but if we turn slightly to look at phagocytosis in neutrophils, we have some different kind of outcomes here. But essentially, when a neutrophil phagocytoses a bacteria or a pathogen, this will result in death of the neutrophil. Okay, so remember that macrophages can be quite long-lived, but neutrophils are very short-lived. And once they've taken up some material by phagocytosis, that results in a regulated cell death. And it's coordinated by various sort of receptor-activated signaling pathways. So first of all, we have apoptosis. So remember, apoptosis is obviously our regulated cell death. And this is induced by tumor necrosis factor that is secreted from natural killer cells and cytotoxic T cells. So there are two types of immune cell that we will talk about in the upcoming lectures. But this TNF will bind the tumor necrosis factor receptor on the neutrophil, and we'll get apoptosis. And this is immunologically silent, so it doesn't kind of further propagate any sort of inflammation or immune effects. We then have pyroptosis, so this literally means fiery death, and this is induced by the inflammasome. So remember we talked about the inflammasome, so we have our nod-like receptor NALP3, and once that kind of forms the inflammasome, we'll get caspase being released, and that will cleave pro-IL-1 beta into IL-1 beta. We also get IL-18 being formed. Now, that's great. So these are sort of inflammatory mediators, but they've got no way of getting out of the cytosol. They're kind of stuck in the neutrophil. So that's really the aim of pyroptosis, where we get breakdown of the cell, and this is immunostimulatory. So by pyroptosis, we get this release of the IL-1 beta and IL-18 that's been generated by the activation of the inflammasome, because it's that NALP3, that nod-like receptor, which is a pattern recognition receptor. It also is utilized by macrophages. So if a macrophage has taken up intracellular pathogens and it can't get rid of them, basically, then pyroptosis is another way that it can kind of expose any intracellular pathogens to the immune system. And then we have these neutrophil extracellular traps, so our nets. So this is netosis that we talked about in the last lecture. And the activation of these actually requires this NADPH oxidase as well as reactive oxygen species. So you can see here we're getting activation of our toll-like receptors, so our PRRs on the surface of the neutrophil. We're getting this phagosome NADPH oxidase, and then we're getting breakdown of intracellular membranes, and we're sort of getting this modification of histones and decondensation of chromatin. And then ultimately, the plasma membrane will rupture, and we're getting release of these neutrophil extracellular traps that are particularly important in netosis. So this is just a diagram, obviously, that you saw on Thursday. So we've got our neutrophils shown in yellow here, our bacteria in red. And then I think probably you can just see the outline here of what was a neutrophil that has obviously undergone netosis and vomited out these chromatin traps. And you can see that kind of ensnaring some of the bacteria just down in the bottom left of the slide here. So what's important to remember is that at the site of infection, we'll have our activated phagocytes, so our macrophages, and these will be producing various cytokines which are helping to establish that state of inflammation. And just to bring you back to the importance of NF-kappa B, so NF-kappa B is really important in regulating multiple aspects of both the innate and the adaptive immune functions, but it really plays a crucial role in mediating the inflammatory responses. And NF-kappa B will induce the expression of a whole variety of pro-inflammatory cytokine genes, and these are cytokines and chemokines. So remember, cytokines are our signaling molecules, and chemokines are a class of cytokine, but they're important in cell migration. And these are the chemokines that you kind of need to know. And you can see that they have localized effects in this middle panel here, but also some systemic effects. So systemic effects will be whole body effects, so across the whole body. So we have IL-1 beta, and we can see that that activates vascular endothelium locally. It also plays an important role in activating lymphocytes. We have our TNF-alpha. Again, this plays an important role in activating the vascular endothelium. We have IL-6, again, important in lymphocyte activation. And then we have CXCL8, and this is actually a chemokine. And this plays an important role in recruiting neutrophils and various other cells to specific sites of infection and also inflammation, and this is something that we'll look at in a few slides' time. And then we have IL-12, which activates natural killer cells and is also important in the differentiation of T cells. Now you'll see here that three of them, this IL-1 beta, TNF-alpha, and IL-6, also have systemic effects. So these are whole body effects. So you can see that IL-1 beta plays an important role in regulating fever, but it also is important in the production of IL-6. And so IL-6 here is also playing an important role in fever, so you can see you kind of got that positive feedback loop. TNF-alpha, also important in fever. And as we just mentioned, IL-6, obviously important in fever, but also it induces these acute phase proteins to be produced. And that's something that we'll look at in a few slides' time. So we have these localized effects, but also these systemic effects. So if we focus on these three chemokines here, so these are our acute phase cytokines. Now, as we mentioned earlier, we want to keep inflammation localized, except in the instance of fever when we actually need it to be systemic. But in order to avoid damage to, the rest of our tissues, we want to keep inflammation localized. But inflammation can have systemic effects because obviously if you think about it, we're getting production of these inflammatory cytokines when our innate immune cells get triggered by their pattern recognition receptors. So IL-1 beta, IL-6, TNF-alpha are these acute phase cytokines, and these help to mediate and establish inflammation. And this is known as the acute phase response. So the acute phase response contributes to inflammation via the production of acute phase proteins. Okay? And these act as kind of effectors. So we have pyrogens that play an important role in raising your body temperature in the instance of fever. They also cause you to be lethargic and sleepy, because obviously if you're poorly, the last thing your body wants you to be doing is kind of running around and being active. It wants you to kind of sit still and let your body heal itself, because obviously when when you've got fever, for instance, it requires a lot of energy in order to maintain that fever. And so this is actually quite an important tactic for fighting infection. We then have the other two cytokines that were shown on that slide. So we've got CXCL8, which is that chemokine, which is important for recruiting neutrophils. Also, just to point out that you'll see here, it's also called IL-8. So that's another confusing thing about the immune system, is that we quite often have alternative names. That's just the way it is. But just stick with one of the nomenclatures, and that'll be fine. And then we have IL-12, which is important in recruiting natural killer cells as well as maintaining that macrophage response. So looking at these acute phase cytokines and what they do, we can see that they have action on fat and muscle. So we get kind of this energy mobilization as well as release of proteins. And this is important because, as I mentioned, fever obviously requires a lot of energy. They also act on the hypothalamus. So here they're increasing that body temperature that's required for fever. And so obviously the desired outcome of fever and that elevated body temperature is that you're going to get a decrease in that bacterial and viral replication. The acute phase cytokines also act on bone marrow. So if you remember, we talked about neutrophils being held in reserve within the bone marrow, and then they're released in response to infection. So we're getting that mobilization of the neutrophils from the bone marrow with the ultimate aim of sort of increasing phagocytosis by that influx of neutrophils into the site of inflammation. They're also having an effect on the liver, and this is where we're getting production of those acute phase proteins that we talked about. So C-reactive protein and mannose binding protein are a couple of examples of that. And the aim here is to get activation of our complement system as well as opsonization. So these acute phase cytokines have sort of a whole broad range of biological activities in order to coordinate the body's response to infection and inflammation. Now, does anyone know what opsonization is at this stage? Anyone heard of it? No? Okay. So that's something we'll talk about when we come on to complement in the next lecture. But essentially, opsonization is an immune process within the body where a variety of proteins are used to tag foreign pathogens, and these are known as opsonins. And by tagging the pathogen, it kind of promotes the phagocytosis and promotes their elimination by phagocytosis. So if we look at the acute phase proteins, so we talked about how the acute phase cytokines, so IL-6 is one here, for example, they have an effect on the liver, and these will result in the production of the acute phase proteins. So you can see here mannose binding lectin, fibrinogen, C-reactive protein. And these acute phase proteins can bind pathogens, but they can't bind host cells. So you can see that they will tag, so an example here is C-reactive protein. It will tag microbes, and then it will act as an opsonin, so it will enhance that phagocytosis, and it will also activate the complement cascade that we'll talk about. So essentially, the acute phase proteins will help to sort of bind pathogens and enhance that phagocytic response. Now, the acute phase proteins can actually be generated and secreted by the liver very rapidly when we've got an infection. And this is obviously in response to these acute phase cytokines. And you can see on this table that we've got a whole range of acute phase proteins. You don't need to know all of these. Just pick one or two. Obviously, this is something you can look at, so remember that you've got the textbooks. If you ever need to, you can refer back to them and look at them. But moving forward with what you need to know, you just need to know like an example of one of these, for instance. But you can see in terms of once we've got an infection, that they're produced very rapidly via the liver, especially C-reactive protein. And this is actually quite beneficial in measuring inflammation, because if you want to check in the serum whether somebody has an infection, you can look at C-reactive protein, and it actually acts as a good indicator of infection if you want to determine that in the clinic. So, as we've already talked about, cytokines have localized effects on the blood vessel, and this helps to support that recruitment of immune cells. So if you remember here, we've got our macrophage, we've got release of cytokines here, we're getting this vasodilation, we're getting permeability of the vascular endothelium, and we're starting to get migration of neutrophils and monocytes across this endothelium, and that's supporting that inflammation. Now, the movement of cells, or the recruitment of neutrophils and monocytes to the affected tissue from the blood, is known as extravasation. And cell adhesion molecules control the interactions between the leukocytes, but also the endothelial cell walls in that inflammatory response. And that's what we'll look at in a little bit more detail now. So just to recap, extravasation is the passage of those immune cells across the endothelium, and it's done in this sort of sequential pattern by activation of surface molecules on both the leukocyte itself, but also the endothelium. And it follows these steps. So we're having chemoattraction, so we're following that chemokine gradient. So if you remember, we were talking about CXCR8, for example. So the leukocyte will be following that like a breadcrumb trail. We then get rolling adhesion, we then get arrest and tight adhesion, and then we get this diapedesis, which is essentially the migration of the cell through the endothelium. So if we look at that in a little bit more detail, but first of all, having a look at these different cell adhesion molecules that are present on both the leukocyte and the endothelial cells. So it's important to remember the three classes. So we've got selectins, integrins, and we've got our immunoglobulin superfamily, or IG superfamily. Now, I don't want you to look at this table and panic, because I realize there's an awful lot going on here. Don't worry, you don't need to know all of this. But another thing to be aware of is you'll see that often we've got a name here, but then in brackets we've got alternative names. So as I mentioned before, it's like that for a lot of the immune system, that things will have different names. So just remember one of them, that's fine. When it comes to this, obviously you need to know the general families, so you need to know your selectins, integrins, and the IG superfamily. And it's good enough just to know one, so you might want to go with P-selectin and LFA1, for example. Okay? Now, the integrins that I've shown here in green, they're present on the surface of the leukocyte, and they play an important role in binding to things like the selectins onto the vascular endothelium. And it's really kind of promoting that leukocyte interaction with the endothelium. Okay? So if we look at the process of extravasation, first of all, we have this rolling adhesion. So we're getting this reversible binding of the neutrophil to the vascular endothelium, and it's kind of rolling over. So you see we've got the flow of blood in this instance. And this interaction is not strong enough to kind of anchor that cell against that force of blood. So we're getting this rolling, and you can imagine like a situation where they're kind of forming almost like a Velcro contact. It's kind of sticking down, but then it's peeling off, and it's rolling over, and it's continuing to stick. But it's causing the cell to slow down effectively on that inner surface of the endothelium and the vessel wall. Now, we were talking about that chemokine gradient, so we've got our CXCL8 that we talked about earlier, and that will start to bind to its specific receptor on the leukocyte or on the neutrophil, and this will trigger the activation of various integrins. So, for example, LFA1 So you'll see here LFA1, it will be expressed on the surface of that neutrophil. At the same time, we've got TNF-alpha, that we also talked about before, that important cytokine, and that will start to induce the expression of various adhesion molecules such as ICAM1 on the vascular endothelium. And what will happen now is that we've got LFA1 on the neutrophil, and this will bind to ICAM1 that's being expressed on the endothelium, and we'll get this tight binding, and this will stop that rolling of the neutrophil. And once the neutrophil has stopped rolling, it's actually able to squeeze through this, the junction in the endothelial cells, and this is kind of the whole process of extravasation. But this step here is known as diapedesis, so the actual kind of squeezing of the neutrophil or the leukocyte through that endothelium is done via diapedesis. Now, we talked about TNF-alpha because obviously it plays an important role in kind of recruiting cells across the endothelium. And it's important that it kind of mediates that local containment of infection, but it can also induce shock within the body when it's released systemically. So you can see here we've got localized infection, and then on the right we've got systemic infection. So TNF-alpha, it's being released by our macrophages, and the aim of that is to induce those local protective effects to sort of recruit those neutrophils. But because it acts on blood vessels, so you can see here that it's obviously it's important in vasodilation and increasing blood flow, and also we're getting that increased permeability to various fluids and proteins in cells through the process of extravasation. That's great, but when we have a systemic infection or sepsis, and TNF-alpha is being released into the body by the macrophages, then we can start to get basically shock within the body. So we start to get this coagulation, we get depletion of clotting factors. This can result in bleeding and potentially organ failure, and also, ultimately cell death. So it's really important to kind of keep these inflammatory events localized, as you suggested earlier. So looking at inflammation, so with the help of Clint Eastwood, really it's the good, the bad, and the ugly. So we have our inflammatory triggers, which are the PAMPs and the DAMPs that will be recognized by the pattern recognition receptors. So they'll be recognizing that injury, that stress, and we'll get the induction of the inflammatory response. Now, the physiological purpose of the inflammatory response is obviously to defend our bodies from infection, also to promote tissue repair, and also the adaptation, to adapt to stress and hopefully restore our normal sort of homeostatic balance. But there are various pathological consequences. So obviously we have autoimmunity as being one of them. We have this autoinflammatory, so we have inflammatory tissue damage and sepsis. We also get this sort of, if you have inflammation that's out of control, you can get this cycling sort of damage and repair. And obviously if you're getting damage and repair, you can get build-up of fibrosis. So it's essentially a bit like a scar tissue. And obviously that is affecting the normal function of the tissues if you've got this constant damage repair cycle going on. Also, unchecked inflammation and damage can result in transformation. So, we've got an impact there of potential cancer if you've got uncontrolled inflammation. And also, a variety of different inflammatory diseases can be developed. So we were talking last week about things like gout, for example. So we have a little poll everywhere quiz. So if you just want to take a couple of minutes to do that, that should be active now. So again, 6 questions, just to kind of test your knowledge from the last lecture and building up to where we are today. All right, I think we'll start to run through it. So obviously, what does PAMP stand for? And it looks like pretty much everyone's got this, pathogen-associated molecular patterns. So remember that they are detected via the pattern recognition receptors. Now, when it comes to inflammatory inducers such as ATP and urate crystals, what are these examples of? So these are examples of our damage-associated molecular patterns. So you can think about ATP being released from cells where it shouldn't be. We think about the uric acid crystals being deposited in the joints of patients with gout. So they're the damage-associated molecular patterns. Toll-like receptors, they're actually present, if you remember from the lecture, I think, last week. They're found on the plasma membrane, but also the endosomal compartments, whereas the nod-like receptors are solely intracellular. The toll-like receptors can be both. The progenitors of macrophages are monocytes. So remember, we have resident tissue macrophages, but we can get recruitment of monocytes and neutrophils, and those monocytes will then mature into macrophages within the tissues. IL-1, IL-6, and TNF-alpha are our acute phase cytokines, so don't get them confused with the acute phase proteins, which will be like the mannose-binding lectin, for example. And what are the correct steps in extravasation? So it is rolling adhesion, arrestin type binding, and then the dark adhesis where the cell migrates through the endothelium, and then we're getting migration following that chemokine gradient towards the site of inflammation. And so that is it, and that's just the reading for this week, and otherwise I'll see you guys on Thursday.